Hello, everyone. Welcome to session two. Before we start, I would like to thank this session sponsor, CSIRO's Minimizing AMR Consortium, of which the Center to Impact AMR is also a partner. Please take time to visit the AMR Action and Insights Knowledge Hub on the QR using the QR code on the screen, and also follow their LinkedIn page. These web pages will also be available for you to look at the sponsor booth in GatherTown, which is the online networking platform we are going to be using for this evening's networking session. And the link to access GatherTown will be sent out shortly in the chat, so keep a lookout for that. And it will be open from the lunch break onwards. And at the end of the session, Charles will give a quick intro in how to use it. So now for our second session, which will be all about the environmental, economic, and social impacts of AMR. And it will be chaired by Dr. Tom Watts and Dr. Gareth Powells. And I will hand over to the session chairs to begin.
Hi, everyone. Welcome back to this session. So as Kalani has said, I'm Tom Watts from Monash. I'm going to be co-chairing this session with Gareth. And this session is all about environmental and economic and also social impacts of AMR. So I just want to remind the speakers that you've got about 10 minutes to talk and a couple of minutes for questions at the end. And for people who want to ask questions, just make sure you use the Q&A function, and we'll get to that at the end of the talk. Okay, so our first speaker today is Muhammad Azreen from Kebangsang in Malaysia, University in Malaysia. And he's going to be speaking about determination of extended spectrum beta-lactamase producing Escherichia coli and Klebsiella pneumoniae carriage in Bangladeshi communities working in the Kiang Valley. Take it away.
All right. So everyone can see my slide, right? Okay, just hope everyone's not sleepy yet. Okay, so I just let's get started. So hi everyone. So I'm Muhammad Azrin, a PhD student at UKM Medical Molecular Biology Institute, Malaysia. So today I'll be sharing on our project on determination of extended spectrum beta-lactamase producing E. coli and Klebsiella pneumoniae in Bangladeshi communities working in Klang Valley. In Malaysia, many migrant workers come seeking better opportunities and wages. They often take on jobs that are less favored by locals but play vital roles in the country economy. While we warmly welcome migrant workers, they may unknowingly carry a pathogen we rather not welcome. In this case, we are looking at the SBR producing E. coli and Klebsiella pneumoniae that which can reside in the guts. So why are these bacteria is actually important? The WHO 2024 pathogen list placed them in the critical group to their resistance to many beta-lactam antibiotics and limited to treatment option. If left unchecked, this could contribute to an estimated 39 million deaths by 2050. So in correlation to Malaysia, the National Surveillance of Antibiotic Resistance has reported a rise in E. coli and Klebsiella pneumoniae infection cases that are already resistant to beta-lactam antibiotics, coinciding with the growing number of our migrant workers. Bangladesh, the second largest group of migrant workers here, has a high prevalence of ESBR producing E. coli and Klebsiella pneumoniae. So while research already suggesting human mobility plays a role in AMR spread, currently there is no data of these migrant workers in Malaysia. So now you might be wondering why actually it's important to know the prevalence of this carriage in this migrant community and what is actually got to do with me. It's actually because the ESBL gene can be silently disseminated to everyone, everywhere, all at once. And it can spread between peoples, animals, the environment, making it crucial for us to understand and monitor it. So we are wonder and we want to know about these three things in the Bangladeshi community. First one, we want to know what is the prevalence of ESBL carriage in the Bangladesh community. Second, what is the anti-migrant profile of this ESBL, EC and ESBL-KP carried by this community? And the last one, what is the most prevalent ESBL genes? So this is how we did it to answer all that curiosity. We begin with the sample collection from Bangladesh community with the help of organization known as the North-South Initiative based on these four inclusion criteria. Then the screening and the isolation were done in the SBL Chrome agar. Those positive growth were identified and subjected to antibiotic susceptibility test by using VITEC2 system. And lastly, by using PCR method, we did the SBL gene profiling to see the prevalence gene in our sample. And now, what actually have we find? And I wonder, are you curious as I am? So let's take a look at the demographic data of our 51 participants first. The majority of our participants are male, and 51% of our participants are aged between 20 to 21 years old. And in terms of working sector, most of them are working in either manufacturing or construction. And majority of them also, Malaysia is their first country of employment abroad. And then the majority of them has been here more than 1 year, and they are sharing accommodation. So from the 51 rectal swab that have been collected, we identified 31.4% of ESBL producing E. coli in our sample, which is a notably high percentage of a carriage. However, no ESBL producing Klebsiella pneumoniae were detected. So since we only have ESBL strain, we later subjected them to the antibiotic susceptibility test using the VITEC2 system to determine their resistant profile. So as you can see here, those in slightly red is a mean resistant, yellow is mean susceptible. So ESBL strain were resistant to ampicillin up to 4th generation of cephalosporin, while maintaining susceptibility to the carbapenems. Regarding to the non-beta-lactam antibiotics, this ESBL-EC show more resistant to ciprofloxacin while remaining susceptibility to others. As you can see on the right side, those with like short form of the antibiotics name, that are the dominant profile that I represent in our sample. So let's say you get an cross-infection from them, hopefully no. This is like the expected antibiogram profile that we can observe. And now let's take a look at the genotype of this strain. Using the PCR method, we perform an ESBL genotype analysis to determine the prevalence of the ESBL gene. So the most dominant ESBL gene that we found in our sample is the BLAST-CTXM1, which is 87.5% of the strain carrying this gene. In terms of the profile, 75% of this ESBL-EC strain only harbored BLAST-CTXM1, while the remaining strain is either carrying BLAST-CTXM1 with additional genes or a variant of CTXM genes. Now, as a conclusion for all finding that we have, we found out that Bangladeshi participants are predominantly male, maybe due to the government program that are really focusing on the manufacturing and the construction works. And based on the literature review, the ESBL-EC poor prevalence in Southeast Asia usually around 27%, and the carriage of ESBL-KP usually low than ESBL-EC, which is mimic our result, which we find out a very high percentage of ESBL-EC with no ESBL carriage detected. And usually, the strain that we collected from community is typically susceptible to more antibiotic. Also mimic with our result where we found out the ESBL-EC from this study still susceptibility to the non-cephalosporin antibiotic, but some show resistance to ciprofloxacin, a very commonly prescribed antibiotic in Malaysia clinics. And then in terms of the ESBL gene, BLAST-CTXM1 is the most prevalent ESBL gene that is consistent with the finding in Bangladeshi data. And it was a gene that first identified in 1986, and it has the most variant globally. And last one, because of the high prevalence of ESBL carrier in migrant worker, it became a significant challenge to the public health and the economy here in Malaysia. So this is the references for further reading. And I would like to say thanks to the AMR detective of our team, which is and also my supervisory team, the grant, the organization, and also the Bangladeshi migrant participant that joined our screening. And I think that's all from me. Thank you.
Thank you. Great talk. We've got a few questions in the chat. So the first one is from Tor, who's asking whether you find any Klebsiella susceptible strains in your sample collections.
Okay, in my sample collection, we have two screening, but in our Bangladeshi community, sadly, we don't found any carriage of ESBL KP, only those participants carrying only ESBL EC only.
Cool. And we've got one more question, which is, can you comment on the strain diversity of ESBL EC that are found in Bangladesh?
Okay, as you can see in terms of the antibiogram profile, basically from the all positive result, the antibiogram profile is sort of similar, which is they are resistant from the ampicillin to the fourth generation of cephalosporin. And then still remains susceptibility to the carbapenem antibiotic. However, there's slightly difference in the ciprofloxacin resistance, maybe due to the taken of antibiotic because ciprofloxacin is a very commonly prescribed in a Malaysian clinic. So it depends on how they take the antibiotics.
I've got a quick question. So I just wanted to ask more about your cohort. So, is this all from a similar dormitory, or are these from multiple different dormitories or
Okay,
different work?
we do a one center study. It's mainly with the help of the North-South organization. They're the one who help us to like calling out all the participant Bangladesh that residing in the Klang Valley. So we are not sure they are come from the same place, the same room or not, but we just only have data they are residing here and what is the type of coordination they are sharing or maybe alone, something like that.
I've got another question if you don't mind, Gareth.
Yeah, can.
So, I was wondering if there's scope to expand this to a sort of a more genomics approach. So to look at maybe more about these isolates themselves. So whether they're clonal, doing some MLST typing or something like that, or some comparative genomics.
Yeah, honestly, this one is the part of our big project. If we have lot of data or a lot of grant, we want to sequence all the strain that we found out. So right now, I already keep it in the glycerol stock for future research. But the main thing now, we only have a look at the ASBL gene. That later we're going to choose a few strain that is quite unique in this community to do whole genome sequencing, just to as a representative from this community.
Beautiful.
Very good. I think we'll move on to our next speaker now. So next we've got Jia Chi from Australia, who's going to talk about the role of mobile genetic elements in nelizolid resistant enterococci and a global and an Australian perspective. So when you're ready, start sharing your slides.
Okay, just before I start, I just want to check if my slide is showing up nice and clear.
All good.
Yep, thanks. Okay, so hi, my name is Jia. I'm a PhD student at the University of Melbourne, based at the Doherty Institute. So my talk today will explore some of the mobile genetic elements, or also known as MGs, in linezolid-resistant Enterococcus species. So Enterococcus species are known to be resistant to a wide range of clinical antibiotics. And because of this, it has been recognized as a WHO high priority pathogen. So in particular, we are concerned about species that are resistant to this antibiotic known as vancomycin, also known as VREs, because these species are often very difficult to treat because they are mostly resistant to all of the first-line antibiotics. So this situation becomes even more concerning when the patient becomes resistant to not just vancomycin, but also linezolid, which is a drug that we use to treat VRE infections. So as with many other antibiotics, linezolid resistance can occur due to mutations on the target binding site of the antibiotic. In this case, it is the ribosomal RNA gene in the bacterial cell. But what we are more concerned of was actually the resistance genes, because unlike the mutations, these genes can be transferred from one strain of bacteria to another strain. So in my talk, I will be focusing on this one gene known as OPTRA, which has been reported in Australia, including in several outbreaks that happened recently, and also in other countries. And not just from clinical patients, but also frequently from livestock animals. So the one that I was trying to understand here was how does MGE play a role in the global dissemination of this gene?
We may have lost our speaker. Gia, are you there?
GRA plasmids and the known transposons, which carry this gene from two species of Enterococcus, Enterococcus faecalis as well as Enterococcus faecium, because these two species represent the dominant human species in the hospital environment. So, once I have these plasmid and transposon sequences, I then use these as reference to map against a global set of enterococcal genomes, which I've downloaded from NCBI. So briefly, I've included both assemblies from GenBank as well as pad and reads from SRE, and then run every tamer which uses AMR Finding Plus to look for the OPRA gene from the assemblies. I've also run MLST to predict the sequence type of these isolates, and finally remove any redundant genomes before reaching a final genome dataset of about 560 genomes for both species. So once I've got these genomes, I then map them to the plasmid and transposon references earlier using Minimap2 with an assembly-to-assembly approach. So from the alignment, we found that two OPTRA backgrounds were dominant in the E. faecalis species. One of these was transposon, known as TN6674, which has been characterized previously, and the other was a plasmid that was about 8.3 kilobase long. So what I found most interesting here was that when we compare the geographical versus the sequence type distributions of both of these backgrounds, we found that the backgrounds actually overlap stronger with the sequence types than the geographical origin. So as you can see here, certain backgrounds such as the transposon are more common in certain STs compared to other STs. So this actually suggests to us that different OPTRA backgrounds
We'll just give Gia's internet some time to start working again, but we might need some extensive questions in the chat if people are missing anything.
Maybe it's best we move on to the speaker for the moment, and then we'll come back to Gia if possible. I'm not sure if she's just entered.
Oh, hi, sorry.
That's all right. Hi, Gia, you're back.
Hi, sorry, yeah, I think my connection is not that stable here. So, yeah,
Try again.
should I share my screen?
Go
Yep,
for it.
we've got some
Okay,
time,
maybe I'll.
so you'd like
Hi,
to keep
Gia.
going.
Just a suggestion, you could turn off your video so it might make things a
Okay,
bit
yeah.
easier on your end. Yeah.
Good idea.
All right, I'll do that. Okay, I'll reshare my screen. This one. Okay. Just want to know, where did I stop just now? Where did I get cut off?
I don't think we saw this slide yet.
Okay. Oops. So
Yep.
this one.
Yep, you're all good here.
Yep. So yeah, in my last slide, I've described an OPTRA plasmid, which has been dominant in the E. faecalis isolates. So we know very little about this plasmid. And from the gene annotation of this plasmid, we know that it carries a multi-resistant unit, which has three antibiotic resistance gene. One of these being the OPTRA gene. So from the literatures, I found that this OPTRA context on this plasmid actually resembles the evolutionary model proposed for the constant resistance gene, MCR1, where the gene was thought to first integrate into a transposon, which then inserts into different plasmid types before spreading in different clones of gram-negative species. So based on that, I established a similar hypothesis that the multi-resistant OPTRA unit from this plasmid could once been part of a transposable unit, and that over time it has integrated into diverse plasmid types with the aid of insertion sequences. And to test my hypothesis, I align the multi-resistant plasmid sequence against other Enterococcus plasmids, and then combine the individual alignments into a coverage plot, which looks something like this. So this figure shows us the coverage plot for the E. faecalis plasmids. And out of 54 plasmids, which has the OPTRA gene that we aligned to the reference, we found that up to 23 of these plasmids share a high sequence homology to the reference plasmid. In other words, these plasmids were predicted to have the multi-resistant OPTRA unit. We then also wanted to know if these plasmids are genetically distinct. So for this, we made a plasmid tree, also called a dendrogram, using the plasmid genes, the presence absence of the plasmid genes from E. faecalis. So this tree shows us three different heat maps. The first heat map tells us which plasmid genes are present. The second heat map, which is the most important one, tells us which plasmids actually have the multi-resistant OPTRA unit. And the last two heat maps tells us the rep types or replicon types and the antibiotic resistance genes present in these plasmids. So from this tree, we could see that the plasmids which carry the multi-resistant unit were distributed across the tree, which suggests that they are quite distinct in terms of the gene content. So if these plasmids are distinct, how did they acquire the resistant unit? This is when I suspect that transposable elements, such as insertion sequences, was involved. So what I did next was to align and examine the immediate upstream and downstream regions of this resistant unit. And from the alignment, I found that in all of these plasmids, although just a few shown here, in all of these plasmids, there was an insertion sequence known as IS1216, shown in yellow, that are present on both sides of this resistant unit, which strongly suggests that this unit could have been mobilized between different plasmids with the aid of insertion sequences. So our third and final finding from the study was that we found that multi-replicon plasmids were involved in disseminating OPTRA resistance in both E. faecalis as well as in E. faecium on the right. So just for information, replicon types are generally used to type gram-positive plasmids, including Enterococcus. And in Enterococcus species, most of the plasmids will have only one replicon type. So although the direct significance of these multi-replicon plasmids in OBTRA resistance was unclear, we thought that they might represent an underlying concern for future AMR siblings, given that some of these can actually accumulate multiple antibiotic resistance genes, including genes that confer resistance to last-line antibiotics, such as linezolid. Right, so in conclusion, we applied a bioinformatics approach to study a global data set of OPTRA positive Enterococcus genomes and the different MGEs, such as plasmids, associated with this gene. So future work will look at the MGE properties of OPTRA isolates from a local setting, including from the recent hospital outbreak, as well as from a One Health sampling that's ongoing in Victoria. So with that, I'd like to thank my PhD supervisors and committee, as well as members from my lab, family and friends for the support throughout this project. So thanks, and happy to take on any questions.
Thank you for a lovely talk, and well done managing with the hiccups there. That was really good. So do we have any questions in the chat? I don't think so at the moment. So I've got one to start with. So you noticed that this plasmid, I think, was distributed in multiple sequence types,
Yep.
which suggests that it's transferable in some way. It's quite a small plasmid. I'm wondering if you found any evidence of mobilizations, RETs, things like that.
So it's quite, you mean things like insertion sequences?
No, it's more along the lines of mobilization. So like a conjugative plasmid that may be able to, mobilize the plasmid into a different bacterial strain.
Um, from what I know from the literature, there are conjugative plasmids that carry this gene, but I don't exactly know what is the extent of the role of these plasmids in spreading this resistance exactly.
Yeah. Okay. It might be worth looking at that small plasmid for any origin of transfer sequences that maybe it can use
Yeah.
to jump into other strains. We've got another.
Thank you.
Well, actually, we've got a question in the chat, which is basically the same thing, but do you have any thoughts on mobilization mechanism of the transposon?
The immobilizing mechanism of the transposon. I think this is not quite well studied in Enterococcus species, especially on this transposon. So the transposon that I've described in my slide, am I sharing my slide just now? No. So there was a dominant transposon that we found in Epicalis. So it has been found in many different isolates around like globally, but I don't think there has been many experimental studies to look at how it transposes out and into different strains. So I think that's something that future we could look into experimentally.
Okay, I have another question if there's time. So regarding your multi-rep plasmid, any thoughts on why they have multiple reps? I know it's quite common in gram negatives to escape things like incompatibility and to maintain plasmids. Do you think the same thing's going on here?
Right. So I can't speak for E. faecalis, but in E. faecium, I suspect that multi-replicon plasmids could evolve because of the antibiotic selection pressures in hospitals, because most of the E. faecium genomes are actually sequenced from hospital origins. So I think there is a selective advantage of having these multiple plasmids in the hospital environment so that to help them survive under different antibiotic selection pressures.
Cool. Thank you. And I think we have one last question in the chat from Abiy. Have you checked eFecium's profile in terms of mobile genetic elements compared to E. faecalis? Hospital-acquired eFecium is a recent problem in hospitals.
Okay, so eFlicium's profile in terms of mobile genetic elements. Do you mean the different plasmid types or like in general everything like plasmids and transposons and insertion sequences?
Maybe.
Yep. So short answer to that, I haven't really checked the overall like all the MGEs present in these genomes. Yep, so it's plasmid, IS, and transposon. Yeah, so I haven't really checked or compared the MGEs, all the MGEs that are present in eFecium versus eFecalis in my data set, but I think it would be helpful to double check on that. Yeah.
Okay, thank you very much.
Right.
Wonderful.
Thanks for the question.
We'll leave it there. So let's move on to our next speaker.
So our next speaker is Anjana Kalum, who's going to be talking about the prevalence of antibiotic resistance in surface water, groundwater, and sediment in the transition zone of the Kalani River Basin in Sri Lanka. So when you're ready, take it away and share your slides.
Okay, can you see my screen and I'm audible?
I don't think we can see the slides just yet.
Oh, there they are.
Oh, it's okay. Okay, thank you very much. Hello everyone, I am Anjana Kalu at the Center for Water Quality and Research, University of Srijayanapura in Sri Lanka. Today I am going to present some of part of my study for visual. So my research topic is prevalence of antibiotic resistance and sorry. Antibiotic resistance is surface water, groundwater, and the sediment in the transient zone of the Canberra Basin, Sri Lanka. So let's start it. Introduction part. Antibiotics are medicines that used to treat bacterial infection by killing bacteria or inhibit their growth. However, when it comes to overuse and misuse of this antibiotic, which lead to increased electroporation on the aquatic environment, ultimately it forced to build up antibiotic resistance. The accumulation of antibiotics in aquatic environment threatens the relevant ecosystem and effect of the health, humans, and other organisms. So what is the antimicrobial resistance? Antimicrobial resistance occurs when microorganisms like bacteria, fungi, and other microorganisms are no longer respond to the antimicrobial drugs. So as a result, antimicrobial drugs are rapidly losing their effectiveness. So according to statistics, AMR is one of the top 10 global health. So AMR projection for 2050, if current trend continues, by 2050, this antimicrobial resistance could kill 10 million people per year. Actually, this value surpassing the cancer value. Therefore, it's essential to determine the antibiotic profile of the environmental valuable area. So in Sri Lankan context, we have selected the Canary Basin as our primary sampling. So the Canary Basin is the second largest, fourth longest and second largest river basin in Sri Lanka, providing significant contribution to fulfill drinking water requirement, especially in Greater Colombo area. So why specifically Canary Basin? It's home for over 25% of the Sri Lankan population, and also it serves as the primary source of drinking water for more than 80% population in the Greater Colombo. And also this river basin help many sustainable industry like pharmaceutical industry, drinking water industry, and there are many industries who utilize this river basin in many different ways. So, significance of this study to identify potential risk, antibiotic contamination and antibiotic resistance lead to critical health issues and or human and ecosystem. And so, therefore, finding identification of the potential is highly important, and also help to understand the role of environmental reservoirs. And also, this is a AMR surveillance regarding this river basin in Sri Lanka. It's provide regional data regarding the AMR. And also, this research will be a foundation for decision making, especially in healthcare sector in Sri Lanka, to prepare a national action plan and update it to combat AMR diseases in Sri Lanka, and as well as fill the knowledge gap, especially in this research sector in Sri Lanka. So during this study, we have covered the 3 UN sustainable goals also, namely good health and well-being, clean water and sanitization, and life on the land. So these are my some research objectives. So methodology part basically divided into 4 parts: water quality analysis, antibiotic resistant analysis, molecular analysis, and quantification of antibiotic genes and residues. At the end of the study, we have planned to conduct NGS and shotgun metagenic analysis to identify AMR gene profile of the whole entire river basin. So here you can see selected antibiotic for this study. There were 10 antibiotics selected for this study, which are belong to 6 major antibiotic class. So here you can see figure number 3 represent major zone of the Canberra Basin. So today I am going to talk about only this yellow color region, transitional zone, because other 2 areas, the sample analysis is still ongoing. That is the reason. So as a sample, total 68 water samples and 33 sediment samples and 16 biological samples are collected. Them predict the total predict this river basin. So after isolation, after collecting sample, we have conduct standard proper method to identify and isolate antibiotic resistant bacteria. After that, we have started and conducted MIC test. Which, initially it was started at 6 p.m. Then we have continued to up to 600 ppm. So here you can see MIC variation of isolated antibiotic resistant bacteria in collected soil sample. When you carefully consider about this figure, you can identify at the initial level there were higher number of antibiotic resistant bacteria isolate available. However, when you gradually increase the concentration of the MIC, so what happened? The number of antibiotic resistant bacteria isolate going to be declined. So similar pattern we have observed for the water sample isolate also. So here you can see table 1 represent water quality parameters of the transition zone of the Canberra Basin in Sri Lanka. The water quality parameters like pH, temperature, nitrate, and conductivity, those parameters are in line with Sri Lankan standard. However, water quality parameters like ammonia and total phosphate did not in line with Sri Lankan standards. Here you can see figure number 10 and figure 11 represent spatial distribution of those two parameters across the sampling location area. As you can see here, in many sampling locations, those two parameters recorded as a highly elevated values. Spatial distribution of antibiotic-resistant bacteria by their respective antibiotic types. So as I previously mentioned, we have tested 10 antibiotics for this study. Here some concluded version. Figure number 12 demonstrates spatial distribution of total isolated antibiotic-resistant bacteria across the Prasinal zone of the Kandiyo basin. As you can see here, Burugodo area and upper middle Kalinga area, Pugodo area, like highly polluted, highly populated area show the highest antibiotic resistant profile, and lowest goes to Pusalio area, and other remaining area remaining as a moderate resistant profile. So after isolate the antibiotic resistant bacteria, we have then categorized them to antibiotic groups. So larger portion goes to penicillin antibiotic class. Lowest goes to tetracycline antibiotic class. According to the published literature by the National Antibiotic Consumption Record of Sri Lanka, that record mentioned that in Sri Lanka, most commonly used antibiotic class is penicillin antibiotic. So our result also in line with this result. And antibiotic class like tetracycline, the government had the action to take the ban the usage of this antibiotic class because due to the higher resistance and overuse and misuse. So this might be the reason tetracycline show the lowest resistant profile now. So that action might be possible and applicable. You can confirm with this that one. So here you can see antibiotic wise resistant percentage. So lowest goes to tetracycline and ciprofloxacin antibiotic group, and highest value goes to the floxacillin and cephalosporin antibiotic groups. Antibiotics are floxacillin and cephalosporin commonly used for UTI and respiratory related infection. So according to our published literature in our healthcare sector, they have mentioned that those two antibiotics commonly prescribed, and overuse and misuse of and overprescribe of this particular antibiotic might be the reason these two antibiotics show the highest resistance profile. And antibiotics like tetracycline and ciprofloxacin in Sri Lanka, especially ciprofloxacin antibiotic quite expensive. This might be also reason ciprofloxacin shows the lowest resistance, and also tetracycline, like I previously mentioned, this antibiotic, the banned usage of this antibiotic. This also might be the reason those two antibiotics show the lowest resistant profile. So conclusion, this finding informed that safer fishing practices by identify areas with lower antibiotic contamination and it's contribute to sustainable fishing and other activities. And also this is a support to build up development policies regarding the healthcare sector regarding the AMR, and to prepare and update a national action plan regarding the AMR combat. And also this study revealed that the requirement of the through our investigation of the this entire river basin. So we want to acknowledge Center for Water Quality and Algae Research and Elis Fabrics their financial support. And here are some references, and thank you.
Thank you. Whilst we wait for some questions in the chat, I've just got a quick question, and I was wondering if some of the contribution to this antibiotic resistance might be from fishing or agriculture and farming as well, who use antibiotics.
Sorry, I didn't.
I was wondering if you know whether any farming or farmers use antibiotics in this region.
Yes, yes, and then especially in Gurgod area, and there's some agriculture farms available. They are commonly used that tetracycline antibiotics. Actually, the government has taken the action, but it's always not applicable because people are using it secretly. That might be the reason. So when we are sampling time, we particularly observe that geographical location and surrounding details. So and also there are two hospitals also near available. Might be some runoff also contribute to this reason, I think. So, however, to confirm this, we have to wait until receive the HPLC quantification data. Still, I am processing that analysis. After receiving data, I can confirm that one also.
Cool. And we've got another question in the chat, and it's Are you using any metagenomics for screening for the antibiotic resistance?
Yes, yes, definitely. Actually, that is planned to do at the end of this study because initially I'm going to isolate higher MIC isolate, and after that I'm going to test that particular antibiotic resistant for their check their multiple antibiotic resistivity. After that, I can isolate. I can select some isolate because already I have more than 900 isolate. So this is quite expensive for entire study. We are receiving only $1,000 for, so that's why we are selecting some particular responsible isolate to conduct metagenomic analysis also.
Cool. We've got one more question, and it's whether the antibiotic resistance that you find correlates with the use of different antibiotics in Sri Lanka.
Yes, yes, yes. What you particularly asking?
So the question is, are the findings the same with your antibiotic resistance, and do they correlate with the antibiotics commonly in use in Sri Lanka?
What kind of antibiotic you asking, no? The
Yes.
mostly amoxicillin and cloxacillin antibiotic commonly used because before we start the experiment, we have get some advice with the healthcare sector, preparing some meetings and other events. Based on that, we have selected particular antibiotic classes and we have selected 10 antibiotics. With this limited this time limitation and due to the huge work, so we have selected some 10 antibiotics because it's only we have 2 years to conduct and complete this study. That's why we have selected some selected antibiotics.
Cool. And we've got one more question before we finish up for this session, and it's from Kalani, who's asking how water quality affects microbial communities in the environment, and would changes in water quality affect ARB rates?
Yes, that is very quite interesting. I have only conducted the ANOVA and rather correlation test informally, not officially. So I have found that total phosphate and ammonia ion concentration have some significant relation to antibiotic resistance. So I think basically this gives a microbial proliferation because those two nutrients can help to replicate and essential nutrient. No? So those can contribute to that's a particular water quality parameters to enhance this resistance happening. I think to further confirmation this, I have to wait until received seasonal data. Actually, I am plan to collect sample from dry season and wet season. For now, I have only completed dry season sampling. After that, after completing wet season sampling also, I can confirm this whether they have actually a relationship available with water quality parameters. That's quite interesting, and thank you for highlighting that.
Well, yeah, thank you very much. And thank you to all of our speakers in this session. We'll hand it back over to the organizing committee who will let you know what the rest of the plan is for today. Thank you.
Okay, so before everyone goes on a break, I would like to tell everyone to begin setting up their GatherTown. Yeah, begin setting up their GatherTown accounts. So for this situation, we use GatherTown because it's a lot easier for people to network at the same time without talking on top of each other. So at this stage, everyone should receive the email of the GatherTown invite, which is shown around here. And if you haven't seen the GatherTown link, I'll just post it in the chat and access it through there. So after clicking on the link, you should be able to see the website for the GatherTown link, which shows you to put in your names and your avatar design. It's important for everyone to put their full names rather than a nickname, so it's a lot easier for other people to find you. And after setting up your account, you would probably see this grassy area over here where you would spawn. And if you're unfamiliar of how the GatherTown works, you should be see these different information tutorials on how to use this software quite effectively. And after entering the GatherTown hall, you should be able to see the entire map of the network. And here you can pretty much interact with other visitors in the symposium. You could either talk to them indirectly, or you could go to a private area shown here. And these private areas are quite useful if you want to talk much more privately to other people. And over here at the top, you can see the our sponsor booth over here, as well as the poster room at the top. So at this stage, everyone should be unable to access the poster room because we would open them after the symposium, which is around at 5:30 Australian Eastern Daylight Time. And once the poster room opens, any lightning talk speakers should stand next to their poster. And you can tell based on the names that would pop up at the top. So probably stay there for the first 10 minutes for any questions from the viewers. And after that, feel free to network up until the end. If you guys have any trouble setting up your gather town, please let me know. And you can find me by typing my name onto the search bar with the admin at the top. So that's pretty much it from me. So see you guys in 1 p.m. AEDT, which is roughly around 1 hour from now. So yeah, feel free to network or take a long break, up to you.